Okay, let's see. So we got almost everybody. Um, so the first thing I wanted to discuss was a little bit on um, plans moving forward for final exam, final lab practical, and a possible writing assignment. Um, and uh, and you know, uh, the professor-student relationship it is really a dictatorship that's meant to be a joke, but I can do what I want, right? But that said, I would like to make decisions on the timing of exams and so forth, taking into account uh, the opinion that you all have about it. And so first off, so the current schedule has the lab practical. The current schedule. Let's see here. Let me pull up a calendar. The current schedule has the lab practical in 2 weeks. I believe, yeah, on the 7th of May. And the final exam is not scheduled until the 14th of May, a week later. So the lab practical would be the last regular class meeting, and then a week later is the schedule for the final exam. So, and I do want to put together a small writing assignment of some kind. You know, if that's going to happen, I'll get something put together in the next couple of days. So the question is, and no promises, but just something to explore, is if you would be interested, and you can put your opinion on the chat. We can kind of get a feeling for what people think, and on the chat, everyone can see it. How people would feel about having the lecture final exam on the 5th of May rather than the 14th. So that does put it just 2 days before the final lab practical on the one hand, but on the other hand, we could probably get through all the lecture material today and be done with it. So you would have, let's see, about a week, just short of, you'd have about 12 days before that final exam. And so, just kind of from your point of view, on the one hand, yeah, it's a little less time to study. It's closer to the final lab practical. On the other hand, you'd have it out of the way and you can focus on your other classes during finals week, So go ahead and type into the chat, yes or no for the change. And let's just kind of get a feeling. And, if there's an overwhelming opinion one way or another, we might just go with that. So, if I don't see you put something on the chat, I'll assume you don't care. People can't hear me, right? Okay. All right, here we go. I'm fine with that. So please put in an opinion, or if you don't, I'll assume you don't. It doesn't matter to you. Okay, no. All right. Okay. 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 Okay, so it seems like most people, it doesn't really matter. Okay. All right. Okay, so it looks like, there's a mix, but, I think there really are enough people who would rather not see that happen. So let's, we've got several people who would rather not have the change. So let's stick with the original schedule. So we'll stick with the original schedule. The lab practical will be on the last class meeting on the 7th, with details to come. And then we'll stick with keeping the final exam on the 14th of May. All right, so let's do that. So we're not going to make any change there because we would have to have unanimous or close to unanimous, and there are several of you who are not interested in the change. Okay, sounds good. All right, so let's go ahead and get started on snakes again, and learn some wild and wonderful facts about snakes. Oh, one thing, I think I might have mentioned this last time. I can't remember if I did. But I just want to point out, sort of on another topic, there's a guy, his name's Ken, and he works at Trader Joe's, and this guy took this herpetology class long before I was here with my predecessor, back about, he's just a few years older than me, just a little bit older than me. So I think I was in college still, or maybe even late, no, actually I was in high school when he took it. I think it was 85 when he took the class. And he works at Trader Joe's, and then there's this other guy, Keith, who recently retired from the fire department, and they have made it, unfortunately we're not going on this trip to the desert, but they have attended every single trip since the mid 80s, and continued going on the trips when I started running them, with just missed 2. I think one was a funeral and one was a wedding day. The guy actually got married, the fireman, or the fire guy, so they couldn't go those years. But as this guy Ken, who works at Trader Joe's, is the amateur naturalist, and for those of you who are interested in birds, and I may have mentioned this, sorry if I'm repeating myself, but he went to Bonelli Park. That's just the opposite corner over there near school, Bonelli, the reservoir there, to do some birding. And this was about a week ago when we had some kind of cool weather. And I noticed in my backyard a lot of birds coming through, warblers, things I didn't even know what they were. Warblers are tough. But those of you interested in birds, he is an amazing birder. And in 6 hours at Bonelli Park, which is just, I don't know, a few hundred meters from campus, he saw 85 species of birds, which is just kind of mind-blowing to me to see. So, yeah, I guess that's a good spot. He was there on the right day. So pretty cool. Anyway, let's get started on talking about snakes. So now we're going to move into the other major clade of snakes, which accounts for the vast majority of species and family level diversity. So again, we've got Serpentes here, and we've covered the Scolophagians already down here, and now we're going to go into the Elefantophidians, and that's all of these other snakes. And again, Elefantophidia and Scolophagia seem to be good, well-supported clades. It's the deepest split, with the only caveat being this Anomaleporidae, kind of odd one, which if you look back, may be sister to the rest of the Scolophagians, maybe sister to the Elefantophidians, or maybe sister to all other snakes combined. So with the exception of this Anomaleporidae down here, we have 2 good clades that contain the respective families that you can see. Again, Elefantophidians, the vast majority of snakes with incredible diversity. Scolophagians all kind of look similar to each other. We see some big differences in feeding mechanics when we look at the leptotyphlopids and the typhlopids, some of the movies we watched last time. But blind snakes and thread snakes are pretty similar to each other. But these snakes up here, the Elefantophidians, incredible diversity, morphologically, size, behavioral ecology, where they live, habits and so forth, you name it, pretty incredible. So as usual, we'll start with the more basal lineages, and we'll march our way through, and then I'll point out some of the bigger features that we see, broad patterns as we go. So let's start with the Annelidae. So this first family is a monotypic family, one species in the Annelidae. And it's found in the Amazon basin, okay, throughout much of the Amazon basin. This is like you're going to see with several of these basal elethinophidians, you tend to see fossorial or semi-fossorial taxa. Okay. Now, let's see here. Okay. What we see is this is included in that. This is a semi-fossorial species. Okay, so somewhat of a burrower, not a great burrower, but semi-fossorial. It's a live bearer, and this species of snake, what we see when we look at the elethinophidians in general, okay, compared to the scullacophidians. The scullacophidians, as you recall, tend to eat large numbers of small prey. Okay, with a lot of species kind of specializing on social insects like the larvae and pupae of ants. So lots and lots of small prey. When we look at elethinophidians, in general, for elethinophidians, what we see is that these snakes are capable of eating large prey. So they eat large prey less frequently. Okay. Now, and you're going to kind of see this as we move through. In addition, within the elethinophidians, what we see is that the more basal lineages have a tendency, yes, to eat large prey, but the prey tend to be elongate prey, slender elongate prey, things like caecilians, amphisbaenians, and other snakes. Okay? The ability to eat large, bulky prey, large diameter prey, you tend to see in more derived elethinophidians, and specifically one group that we're going to be coming to. So you sort of see this rough trend. Scullacophidians, large numbers of small prey. Okay. Basal elethinophidians, large elongate prey. Okay. Eaten less frequently typically. Okay. So less frequent feeding, large prey, but elongate prey. And then again, later in the elethinophidians, as we get to more derived groups, we see certain changes occur morphologically in the skull that allow them to eat large, bulky prey. Okay, so I'll come back to that a couple times as we go through these groups. All right, so here's the snake. All right, then we've got this one, the Tripidophiidae. Okay, and this is a modestly sized group showing kind of an interesting disjunct distribution, around 34 species. If you look at the range, kind of an interesting disjunct distribution. We have some species down here in the Atlantic forests of South America, these Atlantic forests down here, and then we get some in northern South America, disjunct up into extreme southern Mexico, and then we've got some species here in the Caribbean, on the larger islands in the Caribbean. Okay. They inhabit a diversity of habitats from xeric, this means dry, okay, so desert environments or similar environments are xeric, so dry scrublands, to rainforest species like in Panama and down here in the Atlantic forest. They include primarily terrestrial species, but also some arboreal species, okay? And these do eat some various prey, so it doesn't perfectly follow the elongate prey thing in the more basal taxa. While most of the more basal elethinophidians are capable of eating elongate large prey, but not too bulky prey, these a little bit of a mix. Some of the derived species can eat a little bit more bulky stuff. So, the various species eat fish, frogs, lizards, also birds and rodents. So quite a diversity of vertebrates. They show some interesting defensive behaviors. You're going to see a number of interesting defensive behaviors in snakes. One is to roll into a tight ball and hide the head in their coils. And this is something you'll see in other species. Ball pythons will do this, hence their name. Your pet one probably doesn't do it, but if you find a wild one, it'll do it. And certain others, we also kind of see this in the rubber boas, okay, the northern and southern rubber boa, which also stick their tail up in the air to look like a head. but kind of an interesting anti-predatory behavior. And then they also have this strange, there are a few species of snakes that do something like this, with a spontaneous hemorrhage from the mouth and eyes, so around the eyes, and you see kind of this hemorrhaging. But as far as this mouth plus eyes, it appears to be unique. Kind of this hemorrhaging where blood appears in the mouth, and it's not really clear if it tastes bad or what's going on, but it's thought to be some kind of an anti-predatory behavior. Pretty strange. Here are a few species of these, showing a little bit of the diversity. So again, we've got terrestrial species, we've got arboreal species. Here's one eating, I think that's a lizard that's being eaten there. Okay, and some of these others. Okay, all right. So last time I talked about how incredibly cryptic snakes can be and difficult to find. Okay, and this is a great example. The Xenophilidae. This family just has 2 species in one genus, and both species, each one, is known from one specimen. So it's known to modern science from one specimen of each. Here's one of them, and that's it. So obviously not a lot known about the behavioral ecology. Based on where the 2 individual snakes were found, they appear to be nocturnal. So they were found at night in tropical forests. That's basically what's known about these snakes. And you can see their range. They are restricted. It's the one in yellow here, and then in green where this other family occurs together with it. So all we have is just on the Malay Peninsula. Okay, that's one species, and then the other species over here in North Borneo, and that's it. So, this suggests that there are probably other species out there that just haven't been described yet, haven't been discovered and described, but so far just one specimen each from these regions here. Okay. One thing that is known is that some of the morphology. So we know that they're nocturnal in tropical forests. So I take that back. We do know a little bit more, and that's based on morphology. And this is, just based on the 2 specimens that were examined. What we see is some interesting specializations, and I kind of hinted at this when I talked about Lialis. So if you recall, Lialis is a genus in the Pygopodidae in the Geckodons. It's a lineage of the lineage of legless geckos. And if you go back and look at Lialis, that was the one that had the highly kinetic skull, hinged teeth, and various specializations for feeding on lizards, particularly skinks. And when I showed you that, I mentioned that there are certain snakes, especially in Southeast Asia, that seem to show specializations that suggest a diet of skinks. So it's not known that these eat skinks, but we see these specializations that are also, again, found in some other snakes that we do know eat skinks. And what they are, one is hinged teeth. So this is something that's shared in common with Lialis. Okay. A couple of other characteristics we see is a dennary diastema. So here's the dennary bone. Okay. And you remember, as far as tetrapods go, teeth on the lower jaw, the dennary is the only one that's going to have them. So we've got the dennary here. And then we've got a diastema, and that is kind of a little space here, some spacing and a bit of space between the most anterior and teeth and then the middle and more posterior teeth, which would provide a place to kind of get that skink locked into place. Okay. The hinged teeth would hook into the cycloid scales of the skink. And as the skink is struggling and trying to escape, the teeth are going to bend forward, okay? And then if one slips, it's going to snap back and ratchet and lock back into those cycloid scales again, okay? And then also caniniform dennary teeth. So the teeth, so here's a lateral view, here's a medial view, and you can see that some of these teeth, particularly this anterior one, okay, very large and elongate. So it gets, kind of canine-like. Of course, canines are unique to mammals, but canine-like, so it's termed as a caniniform, having the form of canine, this tooth of the dentary. So you can imagine, a skink in here, diastema, these long teeth in the front, and shorter ones back here, and hinge teeth, all specializations, usually associated with skinks. Oh, here we have thanks for looking that up. So it says Wikipedia, they did find a half-digested skink in one of them. Okay, so they actually do have data on one eating a skink. So an N of 1, but an N of 1's not fantastic, but it's a whole lot better than an N of 0. So we do know that they at least eat skinks, so pretty cool. All right, so you can go back, kind of compare this with the Lialis. Okay, then we've got the next one. Okay, this is the Anomalocalidae. This is another group that is not very well known and a small group. So this is the one, only 3 species, okay, just 3 species in this family, and 3 or fewer specimens of each species. Okay, so field work is something that sounds exciting to you, and you're interested in Southeast Asia. There's a lot to be done. Tough to find these things, but it'd be pretty cool to get some more specimens. Strange skulls on these things, with these kind of very robust, very sort of non-snake-like teeth, kind of a short jaw. This suggests that this animal, looking at its morphology, is probably one that tends to be restricted to elongate prey, but the natural history is really not known. They're small snakes. Here's their range. So they're found on Borneo and on Sumatra. Okay, but yeah, pretty poorly known, pretty obscure, kind of strange little heads on these things. You can see the tongue there, but pretty interesting snakes. Okay, now we'll get into some families that are have a little bit bigger families that have a little bit more known about them. So the first is the Uropeltidae. Okay, the Uropeltidae. And these are also known as the shield-tailed snakes. Over 50 species. They're restricted to southern India and Sri Lanka. Okay, so pretty restricted range to southern India and Sri Lanka down here. They're known to eat a lot of earthworms. Okay, so they will include some other elongate prey in their diet, but they seem to be pretty big on the earthworms. So here's another example of a relatively basal Elefantophidian that seems to focus on elongate prey. Okay, larger than, say, ant pupae and ant larvae. Okay, not huge. Earthworms, I imagine, get pretty big down here, but elongate prey. Okay, this is a live bearer, so you see several families represented by live-bearing species. To remind you, viviparity has evolved over 100 times in squamates. Okay, so here's an example of a family that shows live birth. So, and also, like again, some of these more basal families of snakes, they tend to be fossorial. Okay, so we see several examples of this, and we see some fossorial specializations that tend to show up in other taxa. So nice examples of convergent evolution specialization and convergent evolution with other taxa. They tend to have narrow conical heads. So this is something we've talked quite a bit about, how having a head of small circumference or small diameter is favorable for burrowing because it reduces the energy, the force, and therefore the energy required for burrowing. So this is something you tend to see in a lot of burrowing taxa. A blunt tail. So this is something that's specific to this group. The tail, well, you do see blunt tails in a number of fossorial specialists, but this one has a greatly enlarged scale or set of scales. Here it's one scale, here it's several. So it has this strange looking tail with a flat surface or an enlarged scale, okay, a little bit of variation there, that it uses to form soil plugs in the burrows, okay? So where these snakes live, you have other fossorial and semi-fossorial snakes that will Okay. So what's going on here is they're making burrows, they're going along, and then they use this tail to pack the soil behind them to form plugs, to form a blockage in the burrow to prevent something, reduce the chances of some other animal, some other snake, for example, following them and preying on them. So they form these soil plugs. Pretty cool. Another convergent characteristic we see along with the narrow heads is that especially this one is the burrowing mechanism. So I mentioned when we talk about amphisbaenians, I compared amphisbaenians with Sicilians. So you saw Sicilian burrowing mechanics in some detail. And then I mentioned that amphisbaenians, which show lots and lots of convergent evolution with Sicilians, show a burrowing mechanism that's roughly convergent with that of Sicilians. And that is that you have some part of the body moving within some other part of the body, okay? In amphisbaenians, as well as these snakes, the body and musculature of the body is moving within the skin, within the integument, to help generate forces to ram the head forward and to burrow. So we see convergent evolution in that respect between amphisbaenians and these snakes, specifically with the body moving within the skin. And then if you want to expand it to some part of the body moving in within some other part of the body in general, you can extend this to convergence with Sicilians. Okay. All right. And now we have another one. And here we've got the Cylindrophiidae. Okay. And what we see with these, a modest group of just around 14 species. Okay. And if we look at their range, you can see that they're in Southeast Asia. Okay. And so we've got them in southern China, Hunan, parts of China, Malay Peninsula, down into Indonesia, Celebes, Borneo, so Southeast Asia. Okay, fairly widespread. Here is another one of those basal elephanophidians that seems to focus on large but elongate prey. So Sicilians, amphisbaenians, and other snakes are a big part of the diet of these snakes. Okay? So elongate prey, large, relatively infrequent feeding compared to blind snakes and thread snakes, but elongate. Okay? These also are live bearing. Okay. Also in Sri Lanka. Okay. You know something? Let me look something up very quickly here. Yeah, I think this map may be erroneous in that way. Let me take a look at something real quick. Okay. Yeah, they are. So I wanted to confirm that. They are, in fact, found in Sri Lanka, as it says here, but the map, which is out of the book, seems to have missed that. So yeah, so a strange disjunct distribution. So this family is also found on Sri Lanka, but not India, not mainland India, just on Sri Lanka. So kind of an interesting disjunct distribution there. They also have so viviparous, and they also have a flattened tail. And this also may be some kind of anti-predatory. You know, here we have one sticking its tail up in the air. May have some anti-predatory function. Okay. This next family it's a sad story, but it's also a very, very cool group of snakes. Okay. And here it's the Bolieruridae. Okay, Bolieruridae. Now, this family is just two species. Okay, just two species, and formerly with a very, very small range, even historically a very small range on Mauritius. Okay, and so that's its historical range. It's only showing the current range now. So the historical range has them on Mauritius, which is a a heavily populated island nation in the Indian Ocean off of Madagascar. And they were on Mauritius, but then later became restricted to a tiny island called Round Island off of the coast of Mauritius. An uninhabited island, 1.7 square kilometers, which is really small. You could walk around this thing, in 20 minutes. You could walk around this island. Again, 2 species, each in its own genus. And one thing that makes this a really cool couple of snakes is that it has a divided the maxillae are divided with a mobile joint, okay? So if you look here at the CT scan, what you can see is here's the maxilla that goes back along here, and you have a articulation here, a joint within the maxilla. Okay, so here's the anterior part, here's the posterior part. So really interesting morphology. A mobile joint, and this is a specialization unique to this family for grasping skinks, for preying on skinks. And in fact, having this a mobile joint within the maxilla is unique among all tetrapods, not known in any other tetrapod. So truly a unique, unusual 2 species of snakes in this family. You can kind of see some of the other, you can see that this one also has quite enlarged teeth in the front, almost caniniform teeth, maybe not quite, but they're enlarged compared to smaller ones back here. You can see the shapes of the jaws and this mobile maxilla. All of these things would suit the snake well to hanging on to those skinks, very slippery due to those smooth cycloid scales. Okay. The one genus, so here we have some old pictures of these things, okay, a nice watercolor and a couple pictures. Bulliuria was last seen in 1975, and it is considered extinct. It's considered fully extinct, and it's largely due to habitat loss, erosion. So the cutting of forests, deforestation, combined with overgrazing by invasive goats and rabbits, destroying the habitat. So this snake is extinct. And this one, Caesarea, is in 1996, the population was estimated at fewer than 250 individuals. With solid recovery efforts, maybe, solid conservation efforts, there may be some possible recovery here, which would be really cool given that is such an unusual animal. And this kind of brings me a little bit to something I think I mentioned previously, and that is, if you have we don't have endless resources, we don't have endless funds, we don't have endless person hours to put into trying to save species, to prevent the biodiversity loss. So you kind of have to make choices as to what you're going to preserve. And the best way to preserve things certainly is to preserve the habitat. Okay, that's number one. So, certain conservation NGOs, like that, I don't want to advertise, but I like the, what's it called? I think it's the Nature Conservancy is one that I really like because they focus on buying up land to prevent it from being developed and destroyed, and that's a really effective way. But you see, different ways to focus and determine, what are we going to save? Well, there are ecological ways to look at things, so-called keystone species that lots of other species depend on because of something a keystone species does. Like elephants are considered a keystone species because they reduced forest cover and keep open lands, which benefit certain other species. Of course, when you try to conserve elephants by putting big fences around and having too many elephants in a given area, then they take down the forest, and that's not good. Take too much of it down. But that's one. Another one that I think is not a good one, just my personal opinion, is the focus on charismatic species that people like to put on postage stamps and stuff and buy stuffed animals of. I mean, that's great. Don't get me wrong, I love pandas as much as the next person, but that's not necessarily the most effective way. And another one is to try to focus on phylogenetic phylogenetically significant lineages. Okay, so if you're given a choice, all right, are you going to focus on saving 10 species that are you going to focus on saving one species that is one of, say, 30 species in a genus that is one of several genera in a big family? Okay, or are you going to focus on trying to save an animal that is in a monotypic family that has been a separate lineage from others for, say, tens of millions of years? Okay, and it's kind of an analogy to that is, if you have a tree, are you saving little tiny twigs on branches that have lots of tiny twigs, or are you going to focus on the twigs where that is the only living twig on a big branch on that tree, and there's only one little twig, it's barely hanging on? You're going to focus on that. And I'm not going to say one's better than the other, but I think it's something to take into consideration to try to preserve that sort of deep lineage diversity in phylogenetic diversity. And this is a perfect example of that. If we lose Caesarea, we've lost this entire family of snakes, very unique snakes. So hopefully some focus will happen on that. Okay, so let's see. I'm going to go back here for a moment with the tree. All right, so, and this family, for example, is thought to be, it's estimated, it's like 90 plus million year divergence they've been on their own. Okay, all right, so now we're going to move more into the boids, and we're going to move into some of these other groups. So this is, it's not perfect with again, with the prey types, but the tendency is, okay, we tend to see some of the more basal elephanophidians kind of focusing on large elongate prey, and then the more derived lineages tend to focus on more bulky large prey. Okay, and one family that includes a number of species that can eat quite bulky prey is this next one, and that is the boids. Okay, Boidae. All right, so Boidae, around 60 species. Quite diverse. So now we're kind of moving into families of snakes that really show much more diversity within the families. We see diverse habitats. We have rainforest species, dry tropical forest species, cloud forest, deserts, such as our rosy boas, our rosy boa in our deserts, temperate coniferous forest, for example, the rubber boas. Okay? Quite diverse in body form. Associated with different habits. So we have terrestrial forms, we have some specialized arboreal forms, aquatic forms like the anaconda. I'll show you a slide of the yellow anaconda, the largest snake, is an aquatic specialist, and also some semi-fossorial species. Okay, so quite a nice diversity. And this diversity is found quite widespread around the world. So we've got the boids in the New World, okay, quite diverse in the New World. This is the rubber boa, by the way, getting up here into Canada. Very diverse, quite diverse in South America, the Caribbean. And then it used to be that boids were exclusively New World, with one exception down here in Madagascar. Okay, but now the iracines, which are the sand boas, have been put in with the boids. Okay, that's the one of the current views, which brings in this part of the range here in Africa, Arabia, and here into India and Southwest Asia. So these are the sand boas here, okay, which I'll show you a couple of pictures of. And we have a nice one upstairs, a couple of them, I think, up in the vivarium. So one from India and one from Kenya. Okay, so, and they have quite varied diets, eat quite a diversity of prey. Okay, and then New Guinea, which is kind of a strange one here, strange disjunct distribution. Okay, so most boids are viviparous. There's like one exception, just one or two exceptions, but most are viviparous, live bearing. Okay, and some of them, not most, but some do have infrared sensitive or heat sensing labial receptors. Okay, so some of them have infrared receptors in the underneath the labial scales, but you can't really see any kind of pits. They simply have the receptors in there. There's no obvious pits like you can see on a rattlesnake has a single pair of pits, or on boas, or I'm sorry, on pythons. But some do have pits associated with these infrared labial receptors. For example, this emerald tree boa here has very well developed pits along the upper and the lower jaw here, and right up front as well, with infrared receptors associated with them. And these are highly sensitive to the heat put off by endothermic prey, and help these animals detect endothermic prey and aim strikes at things like mammals, birds, for example. Let's see here. Here's a little bit of the diversity. So here is a boa constrictor, scientific name, boa constrictor, all right? Rainbow boa, some of or I'm sorry, not a rainbow boa, that's a different one. I can't remember what that one's called. But nice diversity, pretty popular in the pet trade. I think, yeah, here's a rainbow boa up here. Beautiful snakes. I've never dealt with one, but I've heard they're just nasty and they just really big biters, but pretty to look at. So some nice diversity, different body forms, coloration, and so forth. Okay. Here we have a rosy boa, which you're familiar with, and a rubber boa. And then up here we have a couple of examples of these sand boas. So these are the ones that are in Africa and Arabia, India, and they are basically sand swimmers. Okay, so they're sort of semi-fossorial sand swimmers. So they're in loose soil or sand, and they're really good. They stay hidden most of the time, and you can see how the nostrils and the eyes of this snake are set up on top of the head, very dorsally placed, so that animal can sit, rest submerged in the sand or loose soil. Difficult for predators to see, but have its eyes, have its nostrils up there. And what they do is they ambush prey as it walks by. They're big ambush predators. Okay. So the boids include the green anaconda, Eunectes murinus. This is the largest snake in the world, the largest extant snake in terms of mass. Okay, there's another species, a python, that's the longest snake, but this is the largest snake by mass, can get up to around 500 pounds. That would be an exceptionally large individual. These are aquatic species, usually almost always found associated with water. It's kind of got its eyes and nostrils also dorsally set. This is an animal that likes to sit in the water with its sensory organs only exposed, and ambush prey that come to drink by the water's edge. It's a powerful constrictor. Caimans are a big on the menu with these. You know, the largest reliable record is around 25 feet. Okay, and unfortunately, this is another example of you know, how large specimens are getting more and more rare all the time. You know, whether you're talking about sturgeon or sharks or, you know, various other species with indeterminate growth. Indeterminate growth meaning that the animal grows throughout its life, much more slowly when it gets larger than when it was young, but still they grow out throughout life. So if the animal lives long enough, it can get huge. Most fish are indeterminate growers as well. But they're just, you know, like with fish and other indeterminate growing animals, the large specimens are getting rare, very rare, because they just don't last long enough. Crocodilians is another one, last long enough to get that big. Okay, but there are old records of some quite large individuals. This family also, besides including the largest extant snake in terms of mass, also includes the largest snake ever known to have lived, and that is Titanoboa. Okay, some of you may have heard about this one in a previous course. This was found in a Colombian coal mine, little skull fragments, but primarily vertebrae, from 8 specimens, and about 60 million years old. Okay, and it's estimated that it was around over 40 feet long and perhaps up to 2,500 pounds. Okay. And again, the largest extant snake, to about 20 feet. There's a record, like I said, that maybe 25 feet, but, somewhere around there, 550 pounds. And so, I put here exceptional because that is an exceptionally large green anaconda. Here, this is probably more typical size because, if you go out and catch 8 specimens of a given species of snake, it's highly unlikely you're going to get any exceptionally large specimens just by chance. Okay. The exceptionally large ones are rare. So, this is probably a typical size. So who knows how huge this thing actually may have gotten. And then here we've got some examples here. This is in meters. So these are fossils. This is a fossil here. Here is your exceptional reticulated python and your exceptional green anaconda. And here is Titanoboa. There are no reliable records of any extant snake eating a fully adult human. Sometimes children, but no adult human. It seems that they can't get their jaws around the shoulders. Seems to be an issue. However, this thing certainly would have had no problem whatsoever. It would be a snack, in fact. Okay. All right. So, quite a cool family. Okay. So, I'm going to, even though it's not in the PowerPoint here, I'm going to go ahead. Nah, you know what? Let's not. Okay. All right. So next we've got Xenopeltidae. Okay, Xenopeltidae. So this one is a small family. Sometimes called the sunbeam snakes. And Xenopeltids, small family, just 2 species in Southeast Asia. Okay, so 2 species here in Southeast Asia, found in rainforests and also some open habitats. And these are pretty cool because they they have hinged teeth. So here's another example of the Southeast Asian taxon with hinged teeth, which is associated with a diet of skinks. Okay, so hinged teeth that act like little ratchets to help them hang onto those slippery skinks with their cycloid scales. So yet another example of specialization, hinged teeth associated with a diet of skinks. These are semi-fossorial species, okay, and thought to be and they are nocturnal. Okay, so here's the range. Then we've got Loxosemidae. Okay, so we've got a few very small families of snakes. This is a monotypic family, Loxosemus bicolor. That is it. Okay, this one is found in the New World, so it's here found in southwestern Mexico and then down into Central America for the one species. Found in tropical dry forest down there, a semi-fossorial species, and it feeds on various mammals and reptile eggs, including sea turtle eggs. Okay, so kind of an interesting animal. And yeah, back when I was in grad school, we had a faculty member there who had all kinds of crazy snakes. So I got to play with a Xenopeltid. I got to play with a Loxosemid. I'm not bragging, but they are, it was fortunate to be able to play with some of these snakes that he had. Really unusual stuff, difficult to find. I haven't looked. I mean, some of these things might be available in the pet trade with captive breeding, but yeah, very cool. More stuff available now than back in those days. All right, now we've got another family that probably some of you are familiar with. And this is the Pythonids. The Pythonids. About 40 species, exclusively Old World. Okay, exclusively Old World. And so we've got them pretty well represented in sub-Saharan Africa, as well as India, and then down into Southeast Asia and into Australia. We've got several species. So sub-Saharan Africa, India, Southeast Asia, into Australia. Okay, they are quite diverse also. So like boids, Pythonids are quite diverse in body form and in habits, with terrestrial, arboreal, aquatic, and semi-fossorial species. Actually, let me fix this. These are not habitats, these are habits. Okay, habits and found in a variety of habitats. Pythonids are all egg layers, and there are a few species that actually incubate their eggs, show some degree of parental care. So parental care is rare in snakes, but here we have one where certain pythons will, the female will stay with her clutch of eggs, and then she will shiver. And it's not true, true shivering is a mammal behavior, something mammals do involuntarily when they're getting too cold for the typical mitochondrial pathway to produce heat, to produce body heat. If you're going to hypothermia, you start to shiver, and what this does is it is your muscles are generating heat as a byproduct of their contractions. Okay, so these are not endotherms, but the females can surround their body, coil their bodies around their clutch of eggs, and do little shivering behavior that generates heat and significantly raise the temperature of those eggs for faster development. Okay, some species have infrared sensitive pits. Okay, so probably the evolution of infrared sensitive pits, at least the pit structures associated with the infrared receptors, is probably independently evolved in pythonids and in boids. Okay, and then again in pit vipers, certainly independently evolved in pit vipers. So we see a few independent evolutions of this to infrared receptors to detect endothermic prey and aim strikes. Okay, so let's look at a little bit of the diversity here. All right, so this is a woma, which is a very basal pythonid from Australia, carpet python, a couple of others. Superficially similar to some species of boids, and there is quite a bit of convergent evolution going on there between boids and pythonids, especially arboreal forms, as you'll see. Okay, so the Pythonidae includes Python reticulatus of Southeast Asia. This is the longest extant snake, and there is a fairly reliable record of a 33-foot specimen. So quite large. Every once in a while they pull out, a 15-footer. I think they got a 20-footer once, in Florida out of someone's basement or something. They discover some giant snake, and this happened a few years ago. There was someone found it was like 18 feet or something, a good size, really big snake, basically living in someone's the crawl space under their house down in Florida. And these are escaped pets that just get either escape or are intentionally released. And that snake apparently was living in the crawl space, eating cats. Seemed to be its primary diet was cats, and then other animals like raccoons and possums and things that might go into your crawl space. And it got quite large eating these things, and they pulled it out. This snake, egg layer, as are other pythonids. When a reticulated python hatches from the egg, it's already about 2 feet long. And these, they're beautiful. They're neat snakes. I do not recommend them as pets. They tend to be I've seen exceptions, but they tend to be kind of bitey, not too friendly. And, you don't want to get bit by one of these things. Okay. Now here's what I think a really cool example of convergent evolution. All right. So here we have, let's see, I wanted to check. Let's see. Okay. And just one moment. Okay. All right. So this is a pretty darn cool, you're probably aware by now that I'm a fan of convergent evolution. I think it's really interesting. It raises a lot of philosophical questions about, what are the possibilities, for evolution? What can happen? Are there things that can't happen?
What would we see if we found life on other planets, macroorganisms? Would we see things that look similar to what we see here, or would it be completely different? Fun questions to talk about over beers once in a while. So anyway, here's a great example of convergent evolution. So here we have Corallus chinensis, which is a boid found in the New World. This is found in Central and South America and parts of the West. The genus is represented in the West Indies. This is a South American, Central American species. And then we've got this pythonid, Morelia viridis, okay? And it is found in New Guinea and Northern Australia, okay? And parts of Indonesia. So this boid, this is the emerald tree boa, and this is the green tree python. I'm not going to hold you responsible for the common names, but we've got one respective one each. So this is a great example of convergent evolution. So the most recent common ancestor of the boids and the pythonids was probably a fairly ordinary looking, maybe, I'm speculating of course, but brownish, typical looking python boid looking thing that was probably a terrestrial species, a constrictor, and probably not terribly specialized, but from that, we get the independent evolution in each of these families of snakes that are, number one, they are arboreal specialists, okay? They're found in trees. They are arboreal specialists. Birds are an important part of their diet, so there's some convergent evolution of diet, convergent evolution of habit, convergent evolution of habitat. They live in forested habitats. They are arboreal. Behaviorally, they even have the same unique, as far as I'm aware, to these two taxa, posture for perching on a branch. They find a large branch, they drape themselves over it like a hose and sit there with their head. That's convergent evolution of that behavior, the way they sit on a branch. And convergent evolution of color pattern, both green with some white on there. We get convergent evolution of heat sensing pits in this group. Okay, not uniquely to these two taxa, but the convergent evolution of pits within the boids and within the pythonids is carried over here. Okay, and then even some convergent evolution in the ontogeny of color pattern. The young tend to be red or orange or yellow, and then they undergo an ontogenetic change into this adult green coloration. So really a spectacular example of convergent evolution. The same guy back at Berkeley that had all the snakes also had several specimens of each of these. And it was really cool. They set up these tanks and basically they put PVC pipes and glued PVC pipes across the middle of the tank, and the snake would pick one and drape itself there, and that is the only way you ever saw that snake. And then had little misters to keep them nice and mist them once in a while, since they were rainforest species. So spectacular convergent evolution. Okay, so now let's switch gears into the, let me take a look here where we are. Okay. All right, I think I'm going to stop there for lecture. And then why don't we do a little bit of lab? We'll do a few lab specimens. Okay, so I'm going to stop there for lecture, and give me just a moment and I will pull up some lab material here. Yeah, I think all we have is vipers, I believe. Okay, one family left. We're doing well. Okay, so notes. So you should now be able to see the CalHERPS website, and we have our last family of snakes, and that is the Viperidae. The Viperidae, the vipers. All right. The Viperids used to be, it's gone back and forth, used to be split into two different families. The Viperids, which were exclusively Old World, I think with one exception, and then the Crotalids, which were the pit vipers, and those were exclusively New World. So, but now it seems like those have been relegated to subfamilies, and they're all in the Viperids, the vipers. Okay, so let's see. First off, on the vipers. So the only vipers that we have in Southern California are these pit vipers in the genus Crotalus. Okay, the genus Crotalus, which is the rattlesnakes. Okay, and first I want to give you some kind of general characteristics on Crotalus that apply to all of the species in this genus, apply to the rattlesnakes. Let me check. I'm going to check one other thing. Okay. Yep. That's one thing I wanted to check there. All right. So, make sure we're good. Okay. So, rattlesnakes typically are, so rattlesnakes and pit vipers are found only in the New World. Okay, only in the New World. The genus Crotalus is the only genus of vipers that's found in Southern California. Rattlesnakes generally are heavy-bodied. Okay, they're thick-bodied, robust snakes, much more so than for example, Pituophis, the Pacific gopher snake that's often mistaken for a rattlesnake. So they're heavy-bodied. They have a broad head that is very distinct from a much narrower neck. Okay, so the head is very well defined in rattlesnakes. They have vertical pupils. And rattlesnakes all have strongly keeled scales. Okay, strongly keeled scales. Now, rattlesnakes get their name from the fact that they have a rattle on their tail. Okay? And the rattle on the tail serves as an anti-predatory warning structure used to make a sound for warning, okay? A warning against predators. Now, that said, even though they may generally use it as a warning against predators, and maybe not against all predators, because sometimes they're going to want to avoid attracting attention to themselves in certain cases, but even though it's sort of anti-predatory, the original evolution of the rattle on the rattlesnake seems to, one of the dominant hypotheses out there is that the rattle evolved in vipers that lived in areas with large herds of ungulates, okay, with large herds of hooved mammals, like bison. Okay, and if a bison gets bit by a rattlesnake, it's not going to kill the bison, but it certainly is painful and unpleasant, and something that an animal like a bison would prefer to avoid. And the idea is that the rattle evolved, perhaps in the Midwestern U.S., what's now the Midwestern U.S., to serve as a warning to avoid getting stepped on by these large herding hooved mammals. So that's one of the dominant ideas out there, and it seems to be a pretty good one. And now the rattle's been retained even in places where you don't have any of these large hoofed mammals. Okay. In addition, all rattlesnakes are viviparous. Okay, so the genus Crotalus, and in fact, the pit vipers are live bearers. Rattlesnakes typically show male larger sexual dimorphism. A male larger sexual dimorphism. Now, this is unusual in snakes. In most snakes, females attain larger size than males. That's what you usually see in most snakes. Okay? Why would females get larger? Well, that usually has to do with selection for higher fecundity, okay, higher reproductive output. A large female of an indeterminate growing species can produce more young than a smaller female. Okay, so that tends to select for large females. Okay, why would you have large males? Well, most of the time, large males, as I've said numerous times before when we talk about sexual dimorphism, large males tends to be associated with some kind of contests or fighting or combat between males for either territorial space or direct access to females. Okay, and rattlesnakes, male rattlesnakes, have an interesting combat behavior where they will, two males will meet up and they'll rear up and lift a lot of the anterior part of their body up off of the, and I think I have pictures of this in the lecture when I lecture about these next time. And they basically, they don't bite each other, but they try to, they sort of grapple, okay, and try to push each other down and pin each other down to the ground. And once one has won that contest, then it's the dominant snake. Okay, and this is thought to have selected for larger size in males, with large males having an advantage in these competitive contests. So in rattlesnakes, the males are larger, probably due to these contests they have for access to females, and this is generally true of most of rattlesnakes, with one exception. And that is the sidewinder. That's the last species we're going to talk about. Sidewinder rattlesnakes, the females are in fact larger than the females are larger than the males, but that is an exception. Okay. All right. So let's move into some of the different species here. So we're going to start off with Crotalus atrox. Okay. And this is the western diamond-backed rattlesnake. Okay. So the western diamond-backed rattlesnake is the largest species of rattlesnake in the West. Okay. There are a couple of species in the East that get bigger. The timber rattlesnake and the eastern diamondback, it can get just enormous. They have a specimen at San Diego Zoo it's mind-boggling how big this thing is. I mean, the head is like 5 inches across. I mean, it's just huge. But in the western U.S., this is the largest species of rattlesnake. And they can get up to around 90 inches, okay? So that's, let's see, that's what, 84? So that's about 7.5 feet long, okay? That is a big, big rattlesnake. That would be unusual to see one that big, but they can get quite large. I've seen some good-sized ones. They have a pattern of diamonds on the body. Okay, there are some other rattlesnakes that kind of have this appearance. Okay, fortunately, unfortunately, you can't have specimens in hand. Okay, fortunately, rattlesnakes, if you don't know what to look for, rattlesnakes can be quite difficult to tell apart. If you know what to look for, they're not that hard. Okay, so let me come back to that. Let me tell you about the range first, and then I'll come back to these characteristics that help you identify this largest rattler in the West. So first of all, the range is southeastern California. Okay, the southeastern California. So they are found in the Sonoran Desert. Okay, the low desert. They are absent from the Mojave Desert. We do not have rattlesnakes in the Mojave Desert. Okay, and so they're very much a desert species in Southern California. If we look at the full range of the species, there it is. Okay. The full range you can see from southeastern California well into Mexico, across southern Arizona, into New Mexico, Texas, all the way over here into Arkansas. Okay, and so a pretty wide-ranging species. Pretty wide-ranging species. Again, the largest species in the West. Okay, so that's their range. Again, importantly, not in the Mojave Desert, and they don't get out to the coast. Okay, they don't get east of the Peninsula Ranges, or I'm sorry, they don't get west of the Peninsula Ranges, so they stop at the Peninsula Ranges. You're going to see another species here pretty soon. I believe it's the next one on your list. Yeah, that is the sister taxon, the sister species to the Western Diamondback, and it kind of fills in this part over here, and the ranges butt up against each other. Okay, we'll come to that one next. So how do you distinguish a Western Diamondback rattlesnake from other rattlesnakes? Well, they have one thing they have, which is kind of seen in some other rattlesnakes. Okay, but they have this raccoon pattern on the tail. Okay, which is very well defined on this species. This is also thought to perhaps be an anti-predatory morphology pattern to attract attention to the tail away from the head with a predator. And this raccoon-like tail that you see on this species, the black and white bands go all the way around the tail, and they're, again, very well defined. Okay, but you're going to kind of see that on some of the other species. What a really good one, and I don't know if I can find something. Let's see, where we can zoom in some. Yeah, kind of need a close-up. Let's see. Okay, this is not too bad. Let's see. Should be able to see the zooming in. Okay, it's a little tough to see, but if you look at a diamondback, a Western Diamondback rattlesnake, a little tough to see. You kind of have to use your imagination. Stop by the lab sometime. You know, next year when I'm hopefully teaching herpetology again on campus, feel free to stop by and take a look at specimens you didn't get to see. You're welcome to do that. What the Western Diamondback rattlesnake has is on its scales, and you just have to take my word for it, they have little black pepper-like spots. It basically looks like someone took a pepper grinder, put it on the fine setting, and put a little scattering of pepper across the scale. So they got these little peppery scales. So if you use the raccoon-like tail, you know where the snake came from, and you see those little pepper-like spots on the tail, those are a good combination of characteristics to ID this species. You can see it a little bit. Again, it's not really, quality's not quite good enough, but you can see the little peppering on there. And that's a pretty good character for this species. Okay, let's see. A little bit on the behavior, the biology. So this snake eats kind of a variety of prey. Young ones will eat lizards, other small vertebrates. As they get larger, they tend to move into mammals and birds. So lots of endothermic prey in the diet of this snake, including some quite large prey. Large adults can eat good-sized rabbits, can eat quite significant rabbits. One thing I'm going to talk about in lecture a bit when we get to this family is that of all the snakes out there, vipers are the champions when it comes to eating prey that is large relative to themselves. The records are held by some old world vipers, where they have examples of mammalian prey over twice the mass of the snake. Okay, so they have very long quadrate bones. I haven't talked in detail about snake feeding in Elefantophidian snakes. I'm going to do that a little bit in lab. But the modifications that allow Elefantophidian snakes to swallow large prey, especially those that eat large bulky prey, are really accentuated in vipers. So rattlesnakes can eat very large prey. You know, here we've got one eating a dove. Actually, now you can see here, you can see on the head, the peppering. Okay, this is skin stretched, and there are the scales. You can see the little peppers on there. So swallowing down a dove, a pretty easy prey item there. Oh no, let's see. Just doing a little search here, see if we can find something, kind of a crazy example. Okay. I think this is an eastern diamondback, different species, but let's see here. Yeah, so here's one eating a nice sized rabbit. Okay. All right, so we should be back here. All right. Okay. And I'll probably repeat this. The timber rattlesnakes back east, they get into Arkansas, other areas. There's some that this guy at the University of Arkansas has been studying for decades. And there's some that eat these huge rabbits, and large adults will eat one rabbit a year. That's their entire food intake annually, is one large rabbit. And he has radio tracked these, documented these, and it's kind of an amazing, extreme eating one meal a year. That's not the case with these. There probably are times when one big rabbit will last a year, but there's a related species back further east that will take down very large prey. Western diamondback rattlesnakes are also quite bold snakes. They will often stand their ground and defend themselves rather than flee. I'll tell you, unexpectedly walking up on a hot Western diamondback rattlesnake can really make you feel like your heart's in your throat. I've done it a few times. Quite an impressive sight. And here's kind of an important fact, relatively important fact on this species. It's arguably, probably the most dangerous snake in Southern California. Okay. This one does not seem to have neurotoxic components to the venom like some other rattlesnakes do. The venom of rattlesnakes is generally a tissue destructive venom that causes lysing of cells, destruction of erythrocytes and other cells, huge amounts of swelling, discoloration, and potential permanent damage to the site, the bite site. That's typically what you see in rattlesnakes. Some rattlesnakes, I'll mention them when we get to them, also have neurotoxic components to their venom in certain populations. That doesn't seem to be the case here. But what makes this snake so dangerous is simply the fact that it can get so big. Okay, and this business, you hear people talking about baby rattlesnakes being more dangerous than adult rattlesnakes because they can't control their venom or the venom's more toxic. That's BS. When it comes to rattlesnakes, for a given species, the bigger the snake, the worse, simply because it has the potential to inject a greater volume of venom. What really seems to be important in determining whether, how dangerous a rattlesnake bite is whether or not the snake actually injects venom. Sometimes a snake it'll be a dry bite and it won't inject any or very little venom. Other times it'll inject a lot of venom. Another thing that seems to be important is how long it's been since the snake last ate. If it just ate a big meal and injected a bunch of venom into a rabbit and ate that rabbit, there's not going to be as much venom stored in those venom glands as if it's gone for weeks without eating. Then the venom glands are full of venom, and that's not good for a potential human victim. Okay. The next snake we'll talk about, and this is this next one we've found at Camp Pendleton several times, really a beautiful snake, is the red diamond rattlesnake. Not the red diamondback rattlesnake, but the red diamond rattlesnake. Crotalus ruber, okay, ruber for red. All right. This snake, its range you can see. So the Western Diamondback was over here, and then when we get on the other side, get onto the other side of the Peninsular Ranges, we get the red diamond rattlesnake. So the two ranges butt up against each other. They don't seem to overlap too much. There may be sympatric. I don't know. Let's see what they say about the range here. Morongo Valley. Okay. Yeah, it doesn't say. As far as I'm aware, they don't seem to overlap their ranges, but they butt up against each other. And this species and the Western Diamondback rattlesnake are sister species. They're each other's closest relatives. Okay, and it can be quite an attractive snake. It's similar to a Western Diamondback rattlesnake. Except that it lacks the little salt and pepper speckling, okay? They often have kind of a reddish rust color to them. They can be quite variable. The Western Diamondback, when we get into the eastern part of its range, you get into New Mexico, they can have colors like this too. This one tends to, some variation, but they tend to be kind of, some have a stronger patterning like this individual, okay? Some are really beautiful. Here's from the Santa Ana Mountains. They tend to have the coon-like tail, maybe not quite as contrasting, but they tend to have that as well. Okay, you can see this one's quite contrasting. All right. And this is a male guarding an uninterested female, it says. Okay, so that's one thing rattlesnakes will do too. Here's a couple mating. Is the male will sit on top of the female and mate for a long period of time, and he's sitting there to prevent other males from getting to the female. So they will mate guard. Let's see, pair mating. So some feeding. Oh wow, here we go. That's pretty impressive. Eating a big rabbit. Okay, some of the habitat shots. They like these. They tend to like rocky areas. You often find them associated with rocky areas. Okay, so how do you distinguish this definitively from a Western Diamondback rattlesnake? Well, one is the range. That can help you if you know where you found it. If you're not so sure where you found it, a good character is that the first pair of lower labial scales are usually divided transversely. Okay, I'm going to go ahead and put that into the chat. So the first pair of lower labials. So the labials are the scales that border the mouth. This is the lower. Okay, the first pair at the front of the lower jaw are usually divided transversely. And if I remember correctly, there was a nice little picture, I hope. Where is it? Okay, let's see. Let's see, buzzing sound. Here we go. Here is a comparison. So here's the differences between the Western Diamondback and Red Diamond. So here are some pictures. Okay, so it talks about the markings being less conspicuous, that sort of thing. Here are the scales. Okay, you know what? Infralabial. Okay, they're calling the infralabial, not labial. You know what? Labial, infralabial. It doesn't really matter. Well, this is the labial scale. But here is the first pair is divided transversely, okay? So you can see on this picture, okay, here is the single lower scale. There is the first labial, okay? It's not divided and it goes down like this. By comparison, the first labial is divided into two in the red diamond rattlesnake, okay? Divided into two, all right? We don't have specimens to look at, but if I wanted you to use this, I would provide some kind of picture or something. This shows nicely. So here's a ventral view, divided in ruber, not divided in atrox. Okay? And that seems to be a pretty good character that holds up fairly well. Okay. In terms of they tend to be less aggressive than the Western Diamondback. It's, there are always exceptions. You find individuals that aren't typical, but the Western Diamondback tends to be a bold, aggressive snake. Crotalus ruber tends to be a much more relaxed snake. We've caught beautiful large specimens we found at Camp Pendleton in past years that did not rattle, didn't even twitch the rattle a single time. Pulled them out of the rocks with a hook, carried them down in a bucket, you put them into a tube so people could touch the snake and look at it up closely. Not a single rattle in some individuals. Whereas I've had Western Diamondbacks that rattle when they see you, they rattle when you're hooking them, they rattle the whole time they're in the bucket without stopping. Okay. If I don't do it, well, you know what? I'll just tell you about it now. I talk about not stopping rattling. So I'm going to take a little diversion here and tell you about the rattlesnake rattle. Okay. So the rattlesnake rattle, okay, is powered by what's called the tail shaker muscle. I'm going to chat that in there. Okay. Tail shaker muscle, which is exactly what it sounds like. It's musculature that would be sitting in here, okay, at the end of the tail, anterior to the rattle, okay? And that tail shaker muscle is a series of little muscle slips with tendons in there. And what it does is that muscle shakes the tail. And if the rattlesnake has its tail pointing up in the air like this one does, if you were to look down from the top at that rattle and get some high-speed video of it so that you could see what it was doing, you would see that it's moving in a figure-8 pattern. Okay? It moves in a figure-8 pattern from the top view looking down at the tip of the rattle. It's moving in a figure-8 pattern. Okay? And this works by that tail shaker muscle. And the tail shaker muscle is undergoing little contractions in the muscle slips in the different parts in a very interesting pattern. The tendons play a role in that they're storing energy, elastic energy. So there's sort of this tail shaker and energy storage in the muscle and the tendons moves that rattle around. Okay. Now, one of the important points about the tail shaker muscle that runs the rattle is that the tail shaker muscle is an extremely, I'm going to text this in, highly oxidative, fatigue resistant muscle, okay? This muscle has a lot of slow fibers, slow oxidative fibers, okay? And lots of mitochondria. I'm texting this in for you. At the expense of contractile filaments. Okay, so I'm texting that in for you. So the tail shaker muscle is highly oxidative. That means that it is depending almost entirely on oxidative metabolism performed by the mitochondria using oxygen, okay? As opposed to highly glycolytic muscle that runs on ATP stored in the muscle that is not directly coming from oxidative metabolism, okay? Don't worry too much about the details on the other end of the highly glycolytic part, but these are highly oxidative muscle fibers. So-called red muscle is what you'll see it, the term that you'll see. If you've taken certain classes, hopefully you've been exposed to this, maybe. But since the mitochondria are the ones that are producing the ATP by oxidative metabolism, you have lots and lots of mitochondria in this tail shaker muscle. It is packed with mitochondria, so much so that it doesn't have a lot of contractile tissue. That is, the filamentous elements like primarily actin and myosin. Certainly it has actin and myosin, it has to contract, but not a lot of it. So it is a very low force muscle. If you have one of these rattlesnakes in a tube so that you can play with it and it's shaking its tail, you can just touch the tail and that'll stop it from shaking. It takes almost no force. So the tail shaker muscle is an extremely low force muscle, but a very, very high endurance, slow fatiguing muscle. And a rattlesnake can literally shake its tail for hours without stopping, okay? In fact, this is one of the, it may be the most, one of the most, if not the most highly oxidative muscle in any vertebrate. It's an extreme case of muscle physiology on the high endurance, low fatigue side among vertebrates, a real extreme. So that's pretty cool. Another word about the rattle, a couple other things that I haven't really talked about. The rattle does not have any loose parts. It's not like a typical baby rattle with some loose parts inside. Rather, it is a series of segments, okay? And it's those segments hitting each other when the snake rattles its tail. It's those segments hitting each other that make the sound. There you go. So there's a red diamond that is rattling its tail. So it would be in a figure 8 pattern if you could slow it down and see it. Okay. I wonder, let's see. Yeah, so here's a YouTube video. You can see it shaking. So it's just these segments hitting each other, okay, that is making that sound. And the segments are very, very low in mass. They're very light. So there's very little mass to be moved by the tail shaker muscle. When the animal hatches, strike that when the animal is born, okay, almost messed up there. When the animal is born, let's see, where does he talk about that? Okay. Breeding. Okay. Oh, there's the geographic range from southwestern California, extends all the way to the tip of Baja, by the way. Let's see here. Okay, yeah, right here. So newborn rattlesnakes have a single button, just one little button, and are not capable of making a sound. So a newly born rattlesnake cannot rattle. About a week, approximately a week after that rattlesnake is born, it sheds. And each time the animal sheds, it adds a new segment to the rattle. So about a week after it's born, it adds a segment to that little initial button, okay? And then it can rattle. It's kind of cute, actually. A little tiny 1 week old rattlesnake rattling. Then it can make a sound, okay? So each time it sheds, it adds a segment, and the segments are added proximally. So here's the oldest segment, and then here's the newest one right up against the body. There's one kind of fused to the body there that's dark, okay? And that's the new segment being added proximally with each one. Now, if you find a rattlesnake that has, see if we can see a young one here. That's, can't really see there. And because the animal is growing, yeah, here's a good one. Okay, so this one has all of its segments. It has the original button and then each one that was added with each subsequent shed. And they get bigger and bigger because the animal is larger. Each time it's shed, it's grown. So they get larger and larger. So it tapers down to the tip. Okay. And people have studied, a good friend of mine actually has studied, adding segments only for this. So what I'm talking about applies to all rattlesnakes. All of this rattle stuff applies to all rattlesnakes. And so when you see a taper like that all the way to the button, you've got all the rattles. And what happens is as rattlesnakes get older, this is a pretty fragile structure. It'll tend to get the ends it'll get broken off. It'll get broken off on rocks and so forth. Just climbing around the rocks and doing their usual thing, it'll break pieces off. So older rattlesnakes will have similarly sized segments that kind of end at a large segment because the rest of it's broken off. Okay, and some folks have actually studied rattlesnakes, and what they can do is you can look at the size of the segments, especially with larger snakes. You can look at the size of the segments. You know, when you caught the rattlesnake, you could match it up with rainfall, for example, in past years, and see that the animal doesn't grow much in a poor year. In a rich year with lots of water, lots of prey, it'll grow more. So you can actually measure that. How often do they add a segment? It is possible to age a rattlesnake with the number of segments. If you have a full rattle, none of them are broken off. However, it is not one segment per 1 year. It might be, but it might not be. Okay, and the way you can do it is tricky. You need a big sample of snakes, and you need to basically model. You need a big sample of snakes of known age, and you relate that to where they're found exactly, and you can build a model for a population of snakes so that you can estimate for other snakes. Okay, which isn't too useful for just sort of randomly finding a rattlesnake and trying to estimate its age. And the reason is the rate at which, how often a rattlesnake sheds really has to do with growth rate. And growth rate is affected by environmental factors like, was it a good, rich year? Did the snake get a lot of meals? Was it a long year with, did it stay warm for a long period of time? Or was it a short, cold year without much to eat? And how often the snake sheds is going to determine how quickly segments are added. Okay. All right. So that is some information on rattles. And so let's go back to our rattlesnakes. So that is Crotalus rubra. Oh, one last thing. This is a species of special concern. Let's see here. Yep. Let's see. Species of special concern. California Department of Fish and Wildlife, okay? And it's a species of special concern largely due to habitat loss in the coastal region. So this is an area that's gotten a lot of development, a lot of habitat loss. So it's a species of special concern, which protects it from take with a fishing license. Okay. All right. Next, we have Crotalus helleri. Okay. So there are the next two species, Crotalus helleri. Let's see, that is, okay. He keeps going back and forth. He is not considering them separate species. I'm going to consider them different species, okay? Until I take another look. If there's a new paper, I need to look at that and reassess it, okay? Yeah, he's changed his terminology here. So this is what you'll find in the Stebbins Field Guide. Actually, you'll find Crotalus viridis. It's not the same one. It's the western rattlesnake. So what we're going to do for the next two species is Crotalus helleri, as is shown on your list. So if you look at your list, okay, if you look at your list, you should see that now. We've got Crotalus helleri, the southern Pacific rattlesnake, and Crotalus origanus, the northern Pacific rattlesnake, okay? These two are very similar to each other and generally can only be distinguished by range, unless you can get a genetic sample by where they occur, okay? These all used to be Crotalus viridis, the western rattlesnake, which is what you'll see in your Stebbins Field Guide, okay? So this is the terminology, the scientific and common names that we're going to go with. Okay, so All right. So let's first click on what he's calling Crotalus helleri, what he's calling the, so he's got the common names here, but this would be get rid of the Organus, Crotalus helleri, Southern Pacific rattlesnake. Excuse me. Okay. All right. So Crotalus helleri is the one here that's shown in blue. Okay. So Crotalus helleri is shown in blue. I'm going to kind of talk about these two together and just with a range, really what is distinguishing them. So Crotalus helleri is here in the blue. Okay. This is one that we get. So this Crotalus, neither Crotalus helleri nor Crotalus Organus in red here is found in the desert. Okay. So they're not found in the low or the high desert. Okay. The Southern Pacific rattlesnake is coastal regions of Southern California, going inland some, but not past. It seems to be the barrier here of the San Bernardino, San Gabriel Mountains. It just comes up to the edge, doesn't get into the desert, and then down into coastal regions. Okay, on a couple. It's also found on Catalina. I think this is Santa Cruz Island, Catalina, some of the Channel Islands, and then we get a transition zone here where we move into the Northern Pacific rattlesnake. And as far as Southern California is concerned, we've got it San Luis Obispo County and in Santa Barbara County and Kern County here. Okay, so just northern Santa Barbara. So I would give you information on range where you found this snake. Okay. As I mentioned, the red diamond rattlesnake is found at Camp Pendleton. You do want to know, even though we're not going there, what's found where. Okay. This animal is also found in Camp Pendleton, and it is found on campus. Okay. It's one of the most commonly found snakes on campus. If you walk around for a few hours up on the Voorhees Reserve, there's half a chance you're going to find one of these, or it's going to find you and then let you know where it is when it rattles. Okay. Quite common on campus. Okay, so let's look a little bit at these two. So both of these species have a sort of blotched pattern. Okay, blotched pattern. The tail has black and white bands. Usually, this is an extremely variable. Both of these species are highly, highly variable. Some of them have a little bit of banding on the tail. So you can see, okay, a little bit of banding, pretty weak. I've seen some others with, let's take a look at the Organus. Okay, so I've seen some that have a little bit of banding on the tail, but it's not as extreme black and white as you tend to see in Atrox or in Ruber. Okay. Let's see. But as you can see, just kind of looking through here, an extremely variable. Both of these species are highly variable. They can be very light pattern. Okay. They can be, here's another one with some interesting patterns. They can have sort of diamond-like blotches on them or sort of squarish-like blotches. Here's one that a little more contrasting pattern from Northern California. Okay, here's one from Fresno County. We go back to helleri. Okay, here's one that's from Riverside County that's almost black. It still has some weak patterning on it, but, okay. You know, or you might get this in San Diego County. So quite variable, quite variable. Some juveniles, they can have some really contrasting coloration. There's a nice juvenile there. Okay. So, so sometimes with black and white bands, but not contrasting nearly as much as Crotalus atrox or Crotalus ruber. This one usually has two or more internasals contacting the rostral. Okay, so let's see. Oh, this is a comparison actually between Organus and helleri. Okay, Great Basin. Now this is just looking at what he's considering subspecies. If you look in the field guide, there is in the back cover bit that shows this, but you know what? For our purposes, don't really worry about it. You can take a look. The internasals, those are basically scales that are between the nostrils, and usually this one has more than two of those that contact that most anterior rostral scale. Okay. But you know, we don't have specimens to look at, so don't worry too much about that. This is also a rattlesnake that I kind of like you've seen for a few other taxa we've looked at. You know, this is really one where you figure out that it's not any of the other species, okay, kind of process of elimination. All right. So that's really the best way. Both Crotalus helleri and Crotalus origanus are found in a wider range of habitats than other Southern California species of rattlesnakes. So, you know, you can find them, even though they're not in the desert, in pretty dry areas, woodland areas, grassland areas, quite diverse. You know, you can see that they are quite wide-ranging species in California, okay, with the exception of the deserts. Okay. They include a diversity of prey in their diet. And then something that's notable with this one, and this particularly applies to Crotalus helleri more than Crotalus origanus. I'm not sure that, I don't think there are any populations that this applies to. Let me check real quick to make sure I don't have to correct myself, but I don't think. Let's see what he says. Fangs and venom. Okay. No, I don't see it here. Okay. And then let's take a look at helleri. Huh. Looks like he doesn't talk. Maybe it's somewhere else in the text. Here he talks about the juvenile versus adult thing. Okay. Well, it's known that in some populations, so this is not general for the entire species, but in some populations of Southern Pacific rattlesnakes, okay, the venom has both proteolytic, okay, I'm going to go ahead and just put this in the chat. There you go. Okay, so proteolytic or tissue destructive venom, that is venom that lyses cells and destroys tissue. That is typical of rattlesnakes, as I said before. Okay, this species and one other that we'll talk about in some populations also have neurotoxic components to the venom that affect the central nervous system, do things like depress function of the diaphragm, okay, depress central nervous system function, making them extra dangerous, because now if you're going to the hospital to be treated for snake bite, you're dealing with two very different things: central nervous system interference plus tissue destruction. Okay, so if you go to the hospital for snake bite, it's important for the doctor to know what kind of snake it was and where was that snake found. Okay, and while I'm thinking about it, this is useful information for sort of everyday life in Southern California, especially if you like to go outside a lot. Okay, and that is that. Well, two things. Well, one, if you are bitten by a rattlesnake, you scream your head off at the EMTs or whoever's taking care of you that you want to go to Loma Linda. You start screaming Loma Linda over and over and over again, okay? the world's experts, some of the top world's experts on rattlesnake venom, including that of rattlesnake venom and other types of venom, are a couple of folks at Hayes is one of them at Loma Linda University. If you go to Loma Linda, the hospital at Loma Linda, they will know what to do. They have the best stuff. You will get the best proper care. Okay. Other hospitals, I've had people email me pictures of baby gopher snakes, and they're ready to give the person anti-venom because they think it's a rattlesnake. They don't know what they're looking at, even if the person has the snake or a picture of it. And you really, really do not want anti-venom given to you if you have not been bitten by a rattlesnake. Or if you've been bitten by a rattlesnake and it's a dry bite without venom and you're not reacting, you don't want anti-venom. Okay? There are big implications about it being able to ever use it again, and can be major side effects. It's a big deal. So you only want the antivenom if you need it, and you need the right one. So go to Loma Linda, the hospital at Loma Linda, tell them, put me on the helicopter, get me to Loma Linda. Okay. Secondly, that said, and I think I've mentioned this previously, that said, studies have shown, statistics show that nearly 90% of venomous snake bites in the United States are men between the ages of 25 and 35 years old who've been drinking alcohol, and they get bit on the hands. That's almost 90% right there. Okay. So if you don't fall into that category, I'm not telling you to go pound beers and play with rattlesnakes, but apparently you're less likely to if you're older and female. Okay. Go figure. Human males have a higher risk of accidental death, basically from the time of being able to walk until elderly age, higher risk of accidental death than females, which is kind of interesting because about 50.5% of newborn baby humans are male, 49.9% are female, but by the time you reach reproductive age, it's evened out. Okay? All right. Fun facts from Lappin. It is what it is. All right. So let's take a look here. Next, so that is Crotalus helleri. And, okay. And again, they're not found, I say they're not found in the desert. You may find some at the very, very edge. For our purposes, this animal's not in the desert. They may get just in the edge, right along, very edge, technically speaking. If John's on there, he's probably like, well, I've seen them very edge, but they don't get into the desert like these others, some of these others. Okay. All right, let's see. Next, we've got, a pair of species. I'm going to start with one, and let's see, this is the order on your list. Let me check. Yes, so the next one that we're going to talk about, okay, are these two, Crotalus mitchellii and Crotalus stevensi, okay, the speckled rattlesnake and the Panamint rattlesnake, respectively. Okay, now these two, morphologically, they do have certain characteristics, especially certain color that can help you, but they were considered one species. They were both the speckled rattlesnake in Crotalus mitchellii previously, and then they were split based on genetic data. Okay, and the boundary where one ends and the other begins is kind of a little vague. Okay, the Mojave River, or where it once was, seems to be kind of a boundary. Okay, but with these two, I would give you additional information. Okay, so that is Crotalus mitchellii. And, do they? No. Stevensi. Okay, Crotalus stevensi, oops, and Crotalus mitchellii, sorry. So let's start with mitchellii. Crotalus mitchellii, the speckled rattlesnake. Okay, so actually this shows you very nicely. It shows both of them on here, okay? So Crotalus mitchellii, the speckled rattlesnake, that's in red. Okay, so that one occurs in the southern part of the Mojave Desert, extends down into the low desert, okay, and then also gets out of the desert over here, the western part of the Sonoran Desert, west of the Peninsular Ranges, and out, not all the way to the coast, but it approaches it, okay? So it's out of the desert here, east of San Diego, in San Diego County, gets into Orange County, okay, western Riverside County, and then into the desert, the low desert, and the southern half of the Mojave Desert, okay? Crotalus stevensii, the Panamint rattlesnake, is in the northern half of the Mojave Desert, extending up into the Great Basin. Okay, let's see if he has the full range on here. Yeah, and so this one, yeah, so this is just showing Mitchellii. Okay, so here it is, going down into Baja, southern part of the Mojave Desert, into the low desert. Okay, and then I'm going to switch really quickly to stevensii so we can look at the range. Stevensii in the orange. And then if we look at the full range here, here's stevensii, and it's just in the northern part of the Mojave Desert, gets a little bit into the Great Basin here. Okay? And again, it's genetic material that distinguishes these two. I'm sure that's discussed here. Let's see. Similar snakes found in symmetry. So it's found together with Ruber, Crotalus organis, Crotalus, several species it's in sympatry with. Let's see, information on the two species. Okay, so it gives some of the ground, some of the subtle differences, okay, between the two. Again, these were recognized as the same species until recently. And the colors and patterns are much more variable within each species than anything you can distinguish the two. Okay, so it's really range is the best way to distinguish these two. Okay, so let's start with some more information on Crotalus mitchellii. So Crotalus mitchellii, the speckled rattlesnake, and again, this generally applies to the Panamint rattlesnake as well, has a relatively vague pattern of dark speckled bands. Okay, it usually doesn't have strong markings. Okay, so you can see there are markings, but the boundaries aren't so developed like you tend to see in most of the other species. They tend to have kind of a vague pattern. More than any of the other species of rattlesnakes, both the speckled rattlesnake and the Panamint rattlesnake do a really good job of being cryptic and matching their backgrounds. Okay, and it can be quite remarkable. They tend to have patterns and coloration that are similar to the habitat in which they occur, sort of like this one, like these granitic rocks here. Okay, and then here's a very different one that has kind of this one, the rocks, don't have a strong speckled pattern to them, neither does the snake. Here's the color, very similar. Okay, and these can be, very, very difficult to spot in the wild. You need to be careful. So it can be quite cryptic with their blotched pattern. Okay, they sometimes have a ringed tail, but it tends to be not as obvious as in some of the other species. And if they do have one, the bands, the dark rings are usually narrower than the light rings. Okay, but they tend to have this kind of granitic speckled appearance to them. Okay, again, very good crypsis. Okay, this species occurs both at Camp Pendleton. Okay, the speckled rattlesnake specifically occurs both at Camp Pendleton as well as in the Mojave Desert. And the Panamint rattlesnake, as you saw, that one's just as far as Southern California in the Mojave Desert into the edge of the Great Basin. Okay, let's see. This one, we found this species at the site where we would be, sadly, where we would be camping in the desert. We found lots of these. We found male and female mating with another male hanging out, trying to displace the mating male from the female. And we've had to warn people to be very careful. One time we arrived, people had to go to the bathroom. We just got there, and right there at the entrance to the outhouse, right under the door, was a nice speckled rattlesnake. So we got that one out of the way as quickly as we could. Another time we arrived and we found 3 in the first 20 minutes. So they really like the big granitic rocks out there at that campsite in Mojave. Okay, let's see here. All right, and then the one other thing that I want to mention, let's see, is that one other thing which probably has become pretty obvious here is both of these species heavily favor rocky areas. They really like rocks, and again, they tend to be, their patterns and colors tend to be similar to the rocks in their environment. So if you want to find these snakes, you go to rock outcrops, you go to rocky hillsides, you go to lava flows of rock, and that's the place to really find these snakes. Okay. Next, we've got, so we're down to just 2 more species. Okay. Next, we've got Crotalus scutellatus. Okay. Crotalus scutellatus, the Mojave rattlesnake. Okay. Not the Mojave green rattlesnake. You'll hear that all the time. Okay. Mojave green is not a part of the official common name. Okay. They're sometimes referred to as green rattlesnakes or Mojave greens because they often have a greenish tint to their coloration. Okay. But it's the Mojave rattlesnake. Okay. First of all, if we look at the range of this animal, it is found in the Mojave Desert. Okay. If we look at the full range of this species, okay, if we look at the full range, it's in the Mojave Desert and then gets into Arizona. You see the Colorado River and extends on down into Mexico. But in California, at least, Arizona, it seems to be on the other side of the Colorado River. But in California, at least, it's restricted to the Mojave Desert. We don't get it in the Sonoran Desert, the low desert of California. Okay, so very much in the Mojave Desert. All right, and this animal tends to have quite well-defined blotches or diamonds with light edges. Okay, light edges on them. So on really most of these, so well-defined blotches tend to be very well-defined with light-colored scales forming boundaries around the blotches on the background. Okay, here's another one. They can be a little variable, kind of grays, okay, kind of greenish colors. Here's one, the head, kind of a nice greenish color. Okay, here's an albino. That's kind of cool. All right, so well-defined blotches, typically well-defined blotches. All right, it sometimes has, it often has a ringtail. Okay, however, the bands on the tail are sometimes don't go all the way around. As you can see here, they don't go all the way around the tail the way they do on the Western Diamondback rattlesnake, which is really the key one where this is a good character. So they tend not to go all the way around. The black bands tend to be narrower than the light bands. Okay, like you can see on this one, the black bands tend to be narrower, very well-defined blotches. So those are sort of the characters you can see from a distance. And then if you're able to get close enough, there's a fantastic character in this species that distinguishes it very nicely from all of the others, okay? And that is that it has a single pair of large scales between the supraoculars. Okay, put that in the chat. So the supraocular scales are these large scales, okay, above the eyes, a single large one above the eyes. This is typical for rattlesnakes. Okay, it's typical for many snakes, including rattlesnakes. But then, and there's also a figure of this in the inside cover of your field guide that compares this with others. This is the single pair of large scales. There's one pair between the supraoculars. All other rattlesnakes have more than just the two scales between the supraoculars. Okay, I'm sure that there's probably a diagram in here as well. So the Western Diamondback. Okay, let's see. Okay, so here are the supraoculars here. So you can see there are multiple scales between the supraoculars in the Western Diamondback. Okay, here's another one. Okay, here's a Western Diamondback. Here are the supraoculars, and then multiple scales between them. Okay, whereas in this one, a single pair of scales. And that's a really good character, where even if you have a little pair of binoculars and you can zoom in at one from a distance, you can often pick that out. Okay, so that's a good definitive character for that species. All right, now this one, this is the one other species. So there are two species of rattlesnakes we're talking about that, in addition to the tissue destructive venom, have neurotoxic components to their venom. Okay, the first was certain populations of Crotalus helleri, and the other is some populations of this species. Okay, and in particular, it's in the Mojave Desert. All right, when you get into Arizona, down by the Colorado River in Arizona and going east, you don't seem to see that neurotoxic component so much. But in the Mojave, where you might be out looking around for stuff, they do have a neurotoxic component. Oh, which is one thing that reminds me, one other thing I want to tell you about. In Crotalus helleri, the southern Pacific rattlesnake, okay, I told you that was the one species that occurs on campus. Apparently, those that occur on campus that's one of the populations that includes a neurotoxic component to the venom, okay? Very dangerous animal. The rattlesnakes we have on campus are very dangerous. Don't mess with them, all right? Just look at it and enjoy it, and tell people to stay away, because you'll get a double dose of venom types with those. Okay, let's see, dangerously venomous. Oh, here's one eating an antelope ground squirrel, taking down a nice meal. I've also noticed that, oh, here's some breeding individuals. That's nice, the flowers. I've also noticed that these rattlesnakes are quite. There's one rattling its tail. Okay, so these Mojave rattlesnakes tend to be pretty bold and pretty aggressive, and will really stand their ground and will really, really rattle at you and get up, rear up like this. Like several rattlesnakes will, most rattlesnakes will do this if they're really angry and kind of get up and rear themselves up. So yeah, ones I've come across that were warm were quite aggressive. Okay. All right, and now we're down to our last species of herp. Okay, at least native herps. I'm going to bring out a few non-native things. I plan on showing you a few non-native species, some skeletal material later. I got to figure out how I'm going to do that online. But now we're down to our last one, which is the sidewinder. Crotalus cerastes, the sidewinder. Okay. All right. So, first of all, with the range, we find the sidewinder. And by the way, the official name is not sidewinder rattlesnake. It's just sidewinder. Okay, which I hope is what I have on my list. Yes. Okay. And if we look at the range, oops, wrong one. If we look at the range of this animal, it's found both in the high and the low desert. So both colors, they have two subspecies here. We're not doing subspecies. So they're found both in the Mojave Desert, getting into the edge of the Great Basin, and then down in the Sonoran Desert as well. Okay, so it's very, very much a desert species. Let's take a look at the full range. Okay, so here we have Mojave Desert, Southern Great Basin, the low desert, getting down into Mexico a bit in desert regions. Okay, so very much a desert species. All right, the Sidewinder rattlesnake is, so a couple of things about the Sidewinder rattlesnake. One is it's the smallest rattlesnake in California. Okay, the Western Diamondback is the largest. The smallest is the Sidewinder. All right. In terms, I think I mentioned this, but I think I mentioned this, but if I didn't, now rattlesnakes, as I said, in general, are unusual among snakes because the males are larger than the females. Okay, because the males have combat and so forth. All right. Now, the Sidewinder is an exception to that. Okay, the Sidewinder is an exception to that. Let's see, things in random locomotion, reading. Okay, the females get larger in Sidewinders than the males. The females get larger. Okay, what is that? I'm just trying to find. No, where is it? I want to make sure. Let's see. Let me take a look at this. Yeah, interestingly. So that's interesting. So, he talks about this combat dance. That's when the males rear up to each other and engage in and he does not put that for the Sidewinder. I'd like to check with my friend who's an expert on Sidewinders, but he doesn't talk about that here. So presumably these don't show that combat behavior, the males. Okay, I'm not absolutely certain about that, but I don't think they do, which would make sense with the females being larger than the males. Okay, because you have a lack of that behavior. All right, let's see here. Okay, so in addition, smallest rattlesnake, females get larger than the males. They usually are kind of pale in coloration. They'll have some patterning, but they tend to be paler than most of the other rattlesnakes. There's a little bit of variation in patterns. Here's a very unusual striped individual. This is a mutant basically, but you see cool, weird stuff out there sometimes. Some of them kind of have a reddish color to them. Okay, and the definitive character is that the supraocular scales are modified into having a horn-like structure. They have what look like little horns over the eyes, and that's the supraocular scale modified into a horn-like structure. This is, if you're trying to ID a rattlesnake, this is the first one in your process of elimination. Okay, you look for those supraoculars having the horn-like structure to them. Unmistakable, absolutely unmistakable. Okay. All right. Let's see. Okay. A few other characteristics of Sidewinders. They get their name from the fact that the body moves it in an S-like curve when locomoting. Okay. Let's see. So you can see, and it leaves tracks, little J-shaped tracks like this. And this does a couple of things. These animals, sidewinders, are typically associated with habitat that is has a loose substrate, sandy or otherwise loose substrate. Okay, and you should be able to see this video. And here's a sidewinder sidewinding, and it lifts the front of the body, places it down, then lifts the back part of the body. So it's basically lifting and putting down the front of its body. Okay, with the middle of the body, being lifted off the ground, and this leaves a J-shaped lines. Okay, that point about 45 degrees away from the direction of movement. Okay, don't worry too much about these details, but they point about 45 degrees. So here's that J shape, and so the animal, you can see the direction it's moving. It's moving, up and slightly to the left, but it's pointing at about 45 degrees to the right of vertical. Okay, so that's how you can figure out sort of the direction it is moving. All right, and let's see here. Now the reason it sidewinds is sort of twofold. One is that this is a very effective way to move in a loose substrate. I've mentioned a couple of times that, actually it's probably my other class, but sand in very loose soil has certain properties that make it a little more like a liquid than a solid. Okay, like when you're running on dry sand on the beach, it's acting a little bit like liquid when your feet are sinking into it, which makes you work a lot harder to do your running. That's why running on the beach is such good exercise, okay, especially with bare feet. And so this is a very effective mode of locomotion with that loose substrate. Okay. Another one is that this allows the animal, it's basically walking with its body across the substrate. And by doing that, there's no part of its body that's in contact with the sand or the substrate continuously. Okay. Every part of the body it's taking steps. So every part of the body gets off the sand as another one goes down. So the front comes down, the middle is off. Then the middle comes down, the front and back are off. And that way it can deal with hot sand. If the sand is very hot, okay, then it's able to, even after the sun's gone down, these are nocturnal. Most rattlesnakes in general tend to be crepuscular nocturnal species, okay? And they sit and wait for prey. But this allows the animal to avoid having continuous contact with the sandy substrate. So if it's hot, that's a good thing. So the primary factor is effective movement on a loose substrate that acts like a liquid. Whereas as sort of secondary is that's especially hot. Okay. Another thing that sort of distinguishes sidewinders from other rattlesnakes in Southern California is that sidewinders are big lizard eaters. They primarily eat lizards. Okay, adults will eat small rodents. They certainly will, and other prey, but they are primarily lizard eaters. Whereas the adults of rattlesnakes are mostly endotherm eaters, mammals primarily, and also some birds. Okay, sure, they'll eat a lizard once in a while, but they're primarily mammal eaters. And interestingly, you actually see in most rattlesnakes, you see an ontogenetic change where young ones, newborn and juvenile rattlesnakes tend to eat lizards. And then in most species, they sort of change their diet to eating mammals. And there's even changes in the venom characteristics that sort of go with this, all right? Sidewinders, the smallest rattlesnake in Southern California, even though they'll eat rodents, tend to stick with lizards as their primary diet throughout their life. Okay, let's see, a couple little last factoids on this. I mentioned when I talked about the glossy snake, Arizona elegans, that it was one of 2 species of Southern California snakes that if it's a little bit cold, too cold for snakes to be out at night and you're driving the roads at night and it's kind of cold, it's kind of windy, conditions aren't so good, there are 2 snakes that you're most likely to see when it's just too cold for other snakes to be out. One is the glossy snake, Arizona elegans, which I mentioned when I talked about that one. The other one is the sidewinder. So you might go out on a cold night and see 8 sidewinders and nothing else, but at least you're seeing sidewinders. And then one last little story I'll tell you about sidewinders, and this is actually something that was discovered by a good friend of mine when he was doing his dissertation research back in the 90s and early 2000s on sidewinders. He captured hundreds of these snakes. He did radio telemetry, all kinds of cool stuff. So in general, rattlesnakes, at least some species of rattlesnakes, will show a small degree of parental care where after they give birth to the young, they stay with the young, or the young stay with the mother, okay? The young stay with the mother for about a week until that first shed. Then the young disperse, okay? So in most rattlesnakes, at least in some rattlesnakes, this has been described in black-tailed rattlesnakes, which we didn't talk about, it's an Arizona species. It's been described in this species. I believe it's been seen in the Western Diamondback, where there's some degree of maternal care for about a week until that first shed, after which the young disperse. The cool thing that my friend discovered was he found a hole, okay, with a litter of around something like 10 or 15 newborn sidewinders, okay? And they were all in this tight little ball, okay? And what he observed was sort of group thermoregulation, okay? So you know that ectotherms will thermoregulate. A snake will get out in the sun, get warmed up, go into the shade when it's too hot. A lizard will do this so they can maintain as close as possible their preferred body temperature, okay? Not too hot, not too cold. Now, what he observed was, and he's got great video of this, the juvenile rattlesnakes were in this moving ball, this little ball at the opening to the burrow. And what would happen was you would have whatever ones were on the surface of the ball and in the sun, this is in the morning, would be at the surface in the sun in that ball of snakes. And then they would go down into the ball and others would come to the surface of the ball into the sun for 20 or 30 seconds and then go into the ball. So they were constantly, it was a writhing, moving ball of baby snakes with different ones taking turns getting in the sun and on the surface at the mouth of this burrow, the opening of this burrow. So, and bringing heat into the whole ball. So it was like a moving thermoregulating sort of group thermoregulation in a ball of snakes in these newborn. And this may be something that other rattlesnakes do. It was the first time it was described in snakes were sidewinders that my friend was studying. Okay, that's it for our native species of snakes. That was a pretty long stretch. Wow, almost an hour and a half. Okay, I hope everyone's hanging in there, maybe getting hungry. Okay, and I will, as usual, stay on here for a bit to, if anyone has any questions on the chat. Let me glance to see, let's see. There might have been a couple that I missed. Let me take a look here. Okay, here's.
Yeah, you had a question on Crotalus ruber. Are they primarily a coastal range snake?
Yeah. Okay. So ruber, ruber occurs all the way from the coast up into a little bit into the peninsular ranges, as shown here, and then where they draw, and then down into Baja, all the way to the tip of Baja, and then when we look at Atrox, the Western Diamondback, it kind of picks up where ruber left off, and then it ranges way out to Oklahoma and Arkansas, down into Mexico. Yeah. So, coastal, is, sure, technically speaking, this here is not the coast, but it's, coastal, inland some, but not really getting well into the Sonoran, not well into the Sonoran Desert, not into the Mojave Desert, more kind of coastal regions up to about the peninsular ranges here. Let's see. Okay. All right. So if you came in late, we're discussing a possible change to the date on the final exam, but we're going to keep everything right where it's currently scheduled. You know, I want unanimous or close to it to move anything, and there's several people who would like to keep the dates where they are, and that's just fine. So we will do that. Okay. All right, well, I will hang around for a few in case anyone else has any questions. I'm gonna have to, I don't know, either maybe turn on a fan. I'm gonna try to keep the AC off as long as possible, but it's now 86 degrees in my living room. So, no, I'm sorry, outside, 80 degrees in my living room. Oh, that's nothing. But 86 outside here, probably even warmer when you get downtown a little bit. Unusual hot weather, which is kind of, I guess, unusual hot weather is becoming the norm. And yeah, hope everyone is doing well. I hope that people are hanging in there. I can't remember if I told you, let's see here. Yeah, I think I told you I did some Zoom with, I think I told you in the last lecture that I did Zoom with a good friend and colleague who's from the UK, and his girlfriend has one colleague in his 40s who died of COVID-19. This is in London, and his sister has two coworkers in their 20s, otherwise healthy, who have died of COVID-19. So this misconception some have that if you're young and healthy, there's nothing to worry about, you know, not true. Yeah, and I know there's just another case here in Claremont. Apparently the person was posting on Nextdoor saying that she went to the hospital 3 times with flu-like symptoms, begging to be tested. And they said, well, you don't really have a fever, but you have these other symptoms, but you don't have a fever. And they refused to test her. And I think it was the 4th visit, either the 3rd or 4th visit. She just got really difficult. You know, I hate to say be a difficult person. Sometimes it's called for. Squeak the wheel gets the grease, right? Turned out she is COVID-19 positive. Had to make 3-4 trips to the hospital to get a test. So if the federal government can get its ass in gear and make testing widely available, then I think you should test as many people as possible. But I hope everybody is still healthy, and that we can try to avoid this second wave that's being predicted for the fall and winter. So, okay.
Do you think classes are going to be online in the fall?
Boy, no way to know. It's up in there. We just don't know yet. You know, there was talk that Fullerton had announced this, that they were going online for the fall, but the last email I saw from the Cal Poly, official Cal Poly, is that they haven't confirmed that. So the last I heard is it was actually, I think it was in the newspaper. I think it was in the LA Times a few days ago, but I haven't, as of yesterday, I didn't see good, solid confirmation that that was the case. Yeah, impossible to know. I really hope not, but I think that if we have, you know, if the virus is still at, you know, appreciable levels and there's still a real risk, I wouldn't be surprised if we went all online for the fall. Yeah, I think that's a distinct possibility. No one's really made a call yet. I also think that for faculty that significant furloughs are probably on the horizon and we'll be getting a pay cut, probably of 10 to 20%, which could make it pretty tough for some of the new faculty to cover their mortgages and so forth. But, you know, that's how it is. They did it back in 2000, 2009, I believe it was, with the Great Recession. We got a 10% pay cut, and wouldn't be surprised if it happened again. It's even worse now. And this time, we kind of on top, usually, interestingly, during a recession or during economic recessions, enrollment at universities tends to go up because jobs get harder to come by, so people go back to school. But now we've kind of got it mixed in with this complicating factor of this pandemic. So there's a lot of concern that students, for one reason or another, need to spend time taking care of themselves or family, can't afford it, are not excited about online classes for various reasons. Enrollments might go down, so then that will reduce money going into the universities and make it even tougher to cover costs, including salaries for faculty and staff and grad student TAs and so forth. So it remains to be seen. Just make sure you figure out, with upcoming news, make sure that you know how to vote remotely so that you can vote in the fall. I'm not going to tell you who to vote for, but just do it, because we need leadership like never before. Talking to some really old folks that can remember, the Great Depression, World War II. These are people that are, in their 80s and 90s, and have had an opportunity to really have some interesting conversations with some of these people at the facility where my mom lives, and they're despairing over the lack of leadership at the federal level. They remember FDR, they remember Kennedy, they remember real leaders, giving people realistic comfort and leadership for the states. And that's something that we do not have now. We have quite the opposite, and we need it more than ever. So let's try to get some good people in there. It's fascinating to me how, when you look at some of the states that are starting to open up things early. Georgia, they're going to be opening up movie theaters here on Monday. I think there are going to be certain restrictions in place, but they're going to start opening up movie theaters, tattoo parlors, all these things. And I feel for the business owners. It's brutal, and I think there should be more money going towards helping small business owners. But it's crazy, and it's interesting how there seems to be this correlation between the color of the state and whether it's red or blue, and the talk of opening things up early. Correlations between the level of education in states and talk of opening things up early. It's quite interesting. And I guess, unfortunately, not surprising. It's it sucks. This sucks, but I think it's better than doing things too early and having a second wave, which the Spanish flu back in the 1918, the second wave in a lot of cities was worse than the first one. So I think we need some real leadership. It's interesting how the federal government currently is all about being involved in state affairs when it comes to things like trying to prevent California from having more restrictive emissions laws to prevent the pollution, the air pollution. Ozone, smog, otherwise known as smog, is worse in the LA metro area than any other place in the country. It still is, even with all the emissions in place. And they're trying to relax those rules. But then when it comes to something like helping people during a pandemic, that's up to the states. We can't help you with that. It's just kind of wild to me how that works. Again, just observations. Another one that, and this is another observation that pisses me off about the way things work in this country. And it wasn't this way when I was a kid. It wasn't like this. Things have changed. Of that money, of the I think it was around, I don't know what it was, 400 million, 300 something, 400 million, something like that. That was meant for small businesses and the idea there is to pay people who are not working, pay people who've been laid off so that they can pay their rent and buy food and keep the heat on or the air conditioning or what have you. Okay. That's what that money's for. Off the top of that came around $15 billion for bank fees. The big banks took about 15 billion of that in fees. And the thing that's disgusting about that is normally these fees are associated with making loans that have risk. There's no risk here. The government is giving the money to the small businesses. It's not asking for it back. Okay. They are doing some loans that are basically zero risk loans. The government's going to pay for it. Yet the banks collect $15 billion off the top of that. 15 billion can do a lot for small businesses. I mean, it's just disgusting. And the reason it's like that is because the big banks and the people at the top are buddies with the people in Congress and the president, and they lobby them and they donate to their campaigns, and that's how it works, you know. And it's pretty infuriating. I mean, especially if you're a small business owner. And I have several friends who are small business owners, and they are irate. It's crazy. Yeah. Anyway. Okay, so if there aren't any other questions or anything, let's see. You were saying earlier that we come by and look at lab specimens, classes are in person, and I feel like this might be a stretch, but do you think we'd possibly try to tag along? So the field trips, yeah, it is a stretch. You know, I'd like to say yes. The complicating factor on that is that because of liability issues, undergraduates are generally supposed to be enrolled in a class at the time they're taking the field trip, okay? And other people aren't, you know, I let some other people come along, you know, like John would come now because he is getting units officially for being a volunteer TA, you know, or a couple of grad students or something. But that said, if it depends on the size of the class, you know, I can't really get more than 25 students out there to the Mojave, to these places, either trip. 25 is kind of the limit on total number of students participating because it's simply of logistics, how many cars they allow on the campsite, how many cars they allow in a caravan on the military base, and so forth. Okay. All that said, if the class, you know, this class used to be 25 to 30 students. It's gotten smaller. And the reason it's gotten smaller is because we now have avian biology, fish biology, and mammalogy, all of which were gone for several years. Very fortunately, we have those classes being taught again. So people have more choices, and they can't take everything. So it's getting spread out a bit. Okay, so that's the good news. If the class is a dozen people or 15 people next spring, and there's some space to bring additional people, then yeah, I think there is a possibility that those of you who are enrolled in the class this term, there is a possibility that at least some of you might be able to tag along on field trips next year. And the way I would, you know, if there's not room enough for everyone that wants to go, I would kind of do it on a first come, first serve, and then lottery basis is the way we would do it. But I'd like to say yes, and we'll just have to wait to see. But I think it may be a possibility. It may be a possibility, especially on the Mojave trip where there's a possibility of students even camping in other areas and then tagging along on some of the activities. I think would be a possibility. So I hope so. We will see. Certainly. It's John's birthday today.
I've been exposed.
You've been exposed. So I know you're younger than me, so I can ask, how old are you?
22.
22. That's a good age. Yeah, that's good. Enjoy it. The 20s,
Yeah, I will.
20s are great. The 30s are almost even better. And then after that, yeah.
It goes downhill.
Yeah, it can be good, but when you get into your 40s and your 50s, that's when your metabolic rate really starts to decrease, and your body can't handle some of the physical things that it used to do. Like I was never a great skier, but I was a so-so skier, and I'm simply not going snow skiing anymore. I won't do it because I've seen repeatedly what's happened to friends of mine who are in their early 50s. They tear up a knee or something, and it takes much longer to heal, if ever. You can ask Craig LeMonion about that. He's good now, but it took a few years. It's the aches and pains, just sitting here for a couple hours like this, giving this lecture, I'm going to get up and my back, I'm going to moan and groan and have to stretch a little bit. And that just wasn't the case when I was 22. When I was in my 20s, I could pull all night, and I don't recommend this, and it's not healthy, but I could pull all-nighters, just a little caffeine, and I'd be up all night studying for exams or working on a talk for a conference or something. I could do that well into my 30s. But now, if I get 4 or 5 hours of sleep or no sleep, it messes me up the next day. I feel physically crappy. We talked about the lens of the eye and vision, your hearing, everything starts to go. And all these things that really old people talk about, you start to like, oh, now I kind of get it. It's what they're talking about, except all the time.
Yeah, you realize they weren't lying to you.
They weren't lying. Healing, you heal more slowly. Any sports injuries, you tweak anything, takes much longer to heal. Your memory's not as good. The memory is simply, short-term memory, retaining new stuff is not as good as you get into your 40s and 50s. How much it declines depends a lot on the individual. There's some very old people that have incredible memories, but the memory starts to go, and it's really the way you get by when you start to get around my age is knowledge and wisdom. You know enough that you can kind of figure things out. You know how to go about figuring things out more than when you were younger. But as far as just raw memory power, that declines. If you're dating, people start looking old, and then you got to, remind yourself, hey, a-hole, look in the mirror. You have a picture in your head that you still look the same as you did when you were 30, but you don't. And you got to remind yourself of that when you start getting borderline judgmental on the dating website. You got to be careful. You don't want to be one of those judgmental people.
Can't be too picky at that point.
Can't be
choosers.
You should never be too picky. It's good. Picky's good, but you should never be too picky. But, when it comes to how young someone looks, there's so many interesting things. And I think I probably repeating myself, but all these things that I love to talk about on field trips when we spend hours on the road looking for stuff. But it's fascinating how, you look at online dating sites, and consistently, one thing that I've noticed, I like to drink my beer. I like to have a drink, and one thing I've noticed consistently is that when you see someone online, and I've seen this with women, I'm sure it's the same with men, and you go, wow, that person looks incredible for their age. They really do look 10 or 15 years younger. Inevitably, 95% of the time, a non-drinker, they don't drink alcohol at all. And you see this repeatedly. And you see a difference between even people that drink very moderately, 1 glass of wine a night with dinner, and people that don't drink at all. You see a difference. Yeah, it's interesting. But so be it. I'll take my beer and try to stay young other ways.
Maybe that's why I already have a full beard.
What's that?
Maybe that's why I already have a full beard and I look a lot older than I should.
Oh, you like to drink beer? Well, the primary thing is genetics. It's just, when your hair turns gray, when you lose it, all those things, it's primarily genetics, but then the environment's key, and what you eat matters a lot. Healthy diets, that kind of thing. I've in the past had a pretty bad diet. I do pretty well now. But in the past, you think you can handle anything, It's, well, happy birthday. Enjoy that.
Thank you very much.
Yeah. That's good. Okay, well, I think I'm going to wrap it up. As usual, I'll post this on YouTube so you can see everything. And have a great weekend, and I will see you Tuesday. An article on ResearchGate from this year that describes new specimens of Xenophiidae. Cool. I'll have to update the PowerPoint. All right, I will look that up. Thanks for that. Cool. Check that out. All right.
Hey, do you know what you're doing for their practical?
Not so much. I'm going to have to write a new practical, completely new practical. And I mean, it's basically going to be, some questions will just be in text, that will use knowledge you learned. And then a lot of them, presumably, I know from my other class, the lab and the other course I teach, that you can put pictures and figures and stuff into questions on Blackboard exams. It's largely going to be based on pictures, pictures, maybe range maps, some of that kind of stuff. And I'll probably get the pictures from a variety of sources, but in all likelihood, the primary source will be California herps. So, I've advised students to, study the pictures on California herps, compare find the pictures the best you can, the characters I've discussed. Take a look. We haven't looked a lot at habitat pictures, but take a look at some habitat pictures. I don't expect people to know what all of the different kinds of communities look like, but, if I show you a picture of a coniferous forest and ask you, give you some choices as to species that live there, you would know that the western diamondback rattlesnake is not going to be found in a coniferous forest, but a southern rubber boa may well be found there. Characters, pictures of animals, asking for, a lot of it will be identification, scientific and common names, higher taxonomy, interesting anti-predatory or predatory behaviors, diet, and again, when it comes to things like diet and behaviors, sexual dimorphism, those questions will tend to focus on species that show something interesting or unusual. Conservation status is important, and just like with the previous practical, when it comes to conservation status, you should know a couple things. One, does the species have some level of protection? Two, is it the entire species or just certain populations? And this is indicated on your taxonomic sheet. And three, if the taxonomic status is state endangered, federally endangered, or extinct in the state, you should know that specifically. Otherwise, if it's, threatened or near threatened or whatever, or species of special concern, then you just need to know some conservation status. Okay. Let's see. So
If you need any photos of our SoCal species, just ask me for them.
Okay, cool. And then of course, review the lists that are on Blackboard. It should be complete lists on Blackboard. You should know the specific species that occur on the Cal Poly campus, on Camp Pendleton, or the Mojave, the University of California Reserve and surrounding area. And those lists are on there. I won't be asking which specimens we found on field trips, obviously. That's something I usually like to ask, but I won't be able to do that this time. They probably will be one at a time timed stations. That's most likely how I'll do it. Yeah. And, somewhere between 20 and 30 like 20 stations, each with a sort of 1 to 4 questions. I don't know. I usually like to have phylogenies constructed, and I have an idea for that. Actually, I'll probably email for that. So there's a way to make phylogenies that you don't have to draw the tree, and I will post a Blackboard announcement about that. And that's basically, specimens that are on there will be indicated by likely some kind of picture, and then each picture will have a letter associated with it. So it'll be like specimen A, specimen B, specimen C. So I can say, give the scientific name of specimen A, give the, and so forth. And for phylogenies, normally what you would do is you would have to draw out the tree. The way that you can do that without drawing a tree is you can take the letters and show them with plus signs and parentheses to show the nesting. So if you had A, B, and C, and I was asking for a phylogeny, and A and B were sister taxa with C basal to that, then that would be A plus B with parentheses around it, and then plus C with parentheses around that. And I will post that on Blackboard with a couple of examples so that you can practice doing that. So you're not trying to do it for the first time on the lab practical. So that may be a way that I'll do some testing on phylogenies. Okay. All right. Well, everyone have a good one. I'm going to go ahead and thank you very much. Everyone hang in there and take care. And I'm going to go ahead and first I'm going to save the Zoom. Okay.